Desiccation is a fancy word for drying out or removing water. Here are some examples of some desiccated food items. As you probably will have guessed by now, desiccation is one of the oldest methods of controlling microbial growth in the preservation of food. We still use an awful lot of dried food items. Desiccation does kill microbes. As you remove the water, you're removing water from the cell, and most of them will die. So this is why dried substances will last for a long period of time. And it's only those that are sort of dry, like the raisins off to the side, that you will get organisms that are resistant to osmotic pressure, that are either halotolerant or tolerant of high concentrations of sugars, like what you have in your raisins, are going to be able to survive on it. So that's why things that are dried out have a tendency to go moldy. Fungi, yeasts are osmotolerant. So that's using desiccation to preserve foods by decreasing the number of organisms that are in your food items and preventing other organisms from coming by and eating them before you get a chance to. Desiccation has varying results depending upon the toughness of the organism. Gonorrhea will resist drying for about 1 hour. So you're highly unlikely to get gonorrhea from toilet seats, especially if you make sure they're dry. TB, on the other hand, they have a waxy coat. Remember that mycolic acid, the cell wall of acid-fast bacteria? This prevents desiccation. So even when you're drying sputum that has come onto a table or something like that, you're not drying it, it's just drying. Then the organism resists that drying process and can survive that for long periods of time. Also, to remind you about Chapter 6, we use desiccation as a preservation method for pure cultures. Do you remember what that was? That's right, lyophilization. It's a fancy word for freeze drying. Filtration. This is where you take a fluid or a gas, you run it past a membrane in which there are tiny little holes, we call this a filter, and you physically remove the microbes. It's kind of like what we were talking about in the last chapter when we were talking about filtration to remove the microbes to grow them so we can determine growth. This way we're removing them because we don't want them in either the fluid or the gas. Now, we talk about sterilizing fluids or gases with filtration, but technically this isn't the case because viruses can wiggle through those filters. They're very small. So even though we talk about sterilizing with filtration, we really don't, but it's close enough. Examples of filtration that you may already be familiar with are filtering water, such as if you go backpacking and you filter the water before you drink it. That's getting rid of giardia that our friend the beaver here in the picture is putting into the water. You really don't want giardia. Causes backpacker diarrhea, lasts a long time. Another example of filtration that you may be familiar with is running air through a HEPA filter. HEPA filtration is the standard for sterilizing or disinfecting air in hospitals. We also have a HEPA filter in the lab, and also they've been putting HEPA filters into homes and into vacuum cleaners to cut down on dust mites and all sorts of other nasties that we don't want to be breathing in. Osmotic pressure is related to desiccation. Osmotic pressure is where you're putting a higher sugar or salt concentration outside of the cell, drawing the water out of the cell, and it results in most of them dying unless they're osmotolerant. Now, desiccation, we're removing water to achieve the osmotic pressure. Okay. Osmotic pressure, when we refer to that, that's usually where we're adding sugars or adding salts to something to increase the osmotic pressure. We're doing this without removing the water. Honey is a prime example. This is where we add honey or sugars to substances to make them less palatable to the microbes. For example, if you're trying to preserve your fruits from your garden this summer, what are you going to do? You're going to make jams out of them. Actually, you could make juices and just cap it and can it, but traditionally you didn't have that as an option. So what we did is we added a whole bunch of sugar, and that increases the osmotic pressure, and you end up with preserved fruits. Also hams. If you salt a ham, and hams are all salted, you're adding salt to it, increases the osmotic pressure, makes it so microbes don't live there, and we preserve our foods. Now, we've gotten away from doing an awful lot of this now that we have refrigeration. We don't like things as sweet. We don't like things as salty because we don't have to do that to preserve our food. We still like honey and we still like sweet things, but it's a little less prevalent. Now to the last physical method, using radiation. You're probably going, radiation! Radiation has gotten a bad rap through various events in Japan recently, and also from atomic bombs and what have you. Radiation has gotten a bad rap. But radiation is very useful in controlled ways to control microbial growth. So let's talk about light. Visible light is the spectrum of light, the wavelength of light that you can see, and it does not kill microbes. If we move to shorter wavelengths, more energetic wavelengths, then we start killing microbes. So let's talk about UV. UV radiation is non-ionizing. In other words, it's not removing an electron from an atom, so we're not creating ions. What we're doing is we're creating mutations or bumps in DNA by either denaturing it or by causing bonding between bases that shouldn't be. We'll be talking about this more in Chapter 8, but I want to introduce it here, and I do want you to know that UV radiation is non-ionizing. Now, this is the wavelength that is bactericidal. We use this in microbial biosafety hoods. We put a UV light on it, kills just about everything, and is considered a sterilization method. The problem is it doesn't penetrate. You can use a piece of paper to block UV radiation. Makes you feel better about going into the sunlight, right? So, yeah, if you wear clothes, you're less likely to get burned. Okay. Now, if we move up from that, we're going to skip X-rays because they're not commonly used. If we go to gamma rays, you're probably familiar with gamma radiation coming from outer space, and we use telescopes that pick up gamma radiation to see into distant galaxies. We can also use this to sterilize. Gamma rays are ionizing. Okay. The rays are energetic enough that they're going to knock electrons off of molecules, and we're going to create ions where they shouldn't be. If you create ions in the plasma membrane, then your plasma membrane breaks down. If you create ions in proteins, you change the structure. If you create ions in DNA, you break up the DNA, you cause mutations. Okay, it's sterilizing. Gamma rays penetrate further than UV rays do. So in lab, when we've used those single wrapped pipettes that are in the paper, it's paper on one side, plastic on the other, those have been sterilized by gamma radiation. The radiation goes through, kills the microbes, passes on through, it's no longer there. The item is not radioactive. It's only radioactive if you have an atom that's giving off radiation stuck on there. But when the radiation passes through, it sterilizes, generally does not distort the plastic, and we consider this a method of cold sterilization. We use this when we can't autoclave something and you can't boil it, okay, because it's sensitive to heat or moisture, and we can sterilize powders and all sorts of good stuff. Now, in Europe, they sterilize food by using ionized radiation, by using gamma rays. They wrap the item in plastic, they zap it with gamma rays, and they can put milk and raw meat and all sorts of good stuff on the shelves, and it does never rot. Now, it may break down chemically, and it may go rancid, in other words, but it's not going to rot. It doesn't have microbes in there, and you don't have to worry about E. coli or listeria or all sorts of other stuff. All right, that's it for the first half of Chapter 7. Stretch, take a break, think about something else, come back and we'll talk about chemical control methods. Now for our last subtopic, biosafety levels. The Centers for Disease Control and Prevention, or the CDC, have set up regulations for labs that and research facilities that handle microbes. And this is intended to make sure that people are protected and people outside of the lab are protected. So let's go through the biosafety levels 1 through 4. Biosafety level 1, basically that's our lab here at the school. We're working with non-pathogens or microbes that have a very low virulence. An example would be lab strains of E. coli that we use, Staphylococcus epidermidis, a common skin bacterium that has very low virulence unless you're replacing a hip or something like that. So that would be an example of the organisms that we would use in a biosafety level 1. So you don't have a biosafety hood. You don't have to worry about most people becoming infected with the microbes. And normal dishwashing and medium to low level disinfectants are just fine for this. Biosafety level 2 has you work with organisms that have moderate virulence. So opportunistic pathogens, if you're working with pathogens that aren't opportunistic, but they have a treatment, antibiotics take care of them, that kind of thing, you may want to work with them within a biosafety hood. Examples of organisms that we would use in a biosafety level 2, things like methicillin-resistant Staphylococcus aureus, or MRSA as it is affectionately known. If you have a normal immune system, you're probably not going to become sick with MRSA, but you still want to treat it with respect, working with pathogenic viruses that most people who are healthy can fight off, things like influenza, hepatitis B that we have a vaccine for, you would be able to use in a biosafety level 2. Biosafety level 3, we're working with organisms that are much more virulent. We have high virulence, but generally we have a vaccine for them. Organisms that we would use, that we would work with in a biosafety level 3 would be, for example, anthrax. It can be a cause severe disease, especially if it's been weaponized. Also, you don't want anthrax getting out into farm country where it can decimate your cattle production, but we have a vaccine for it. So that would be an example of a biosafety level 3 organism. Bio-level safety 4, also known as the hot zone, is where we work with organisms that have extremely high virulence. We don't have a vaccine, we don't have a treatment. Ebola, which was the subject of the book called The Hot Zone, we would be working with within a biosafety level 4. Biosafety level 4, you gown up, you have air brought into your suit, you're not ever exposed to the organism, and hopefully there's no chance of it getting out. So here's a summary of the physical methods of control, and here's a reminder of the learning objectives for this topic. Remember to write down any questions that you may have so that we can discuss them in lecture.